Our next panel is about the other three of our Helmholtz Information and Data Science schools, and it's about understanding data from single cells to the great blue sea.
Okay, that sounds exciting. Andreas is already waiting in the back, ready to take over. So I'm going to pass the baton back to him. Goodbye, and thank you so much for the opportunity to host this session.
Bye, thank you.
Thank you.
Thank you, Alex, for the most valuable support, and I hope you guys had fun, right, with our special about women in data science. But I think the most important point is whether you're a guy or a girl or anything in between, when you're passionate about doing great science with information and data science, we're the place to be, and we to support anybody who wants to join us. All right, so I guess we move on. Can I get my seat back? Yes,
of course.
So I was kind of used like leaning on this one. So guys, all right. So we're moving into the next session. Time is flying. So after this very nice session, we will continue our journey through the vast and exciting programs that are called the Information Data Science Academies. But looking back to the last panel, I especially wanted to thank Alice McCarty and Sabine Attinger again, who have been very strong supporters of all the information and data science activities since the year 2016. So if you're still tuned in, thank you again for your years and years of most active support. So I basically introduced this session before when I introduced the first half of the Helmholtz Information and Data Science schools. We will have now a closer look into the three remaining ones, which is the hybrids here in Berlin. Very diverse program. We'll hear about that from Lale Haverdy from the Max Delbrück Center for Molecular Medicine. So Lale, are you with us already? Great to see you. Hello. We will also hear from the MAR data, focusing on marine and coastal. We have with us Anna Bierstock from the GeoMar Center. Anna, great to see you. Hello, good to have you here as well. And we will also hear from the DASH in Hamburg, from Alke Means, from the German Electron Synchrotron DAISY and the XFEL program. Alke, great to have you here as well. So I'm very excited about this session as well. Let's dive right into the introduction of your great schools, and I would like to call the hybrids first. Lale, could you introduce the hybrids to us in a couple of minutes?
Yeah, sure. Hi, happy to be here in this discussion with you. Yeah, I am from Max Delbrück Center for Molecular Medicine in Berlin and MDC. So this is we are also coordinating the hybrids graduate school, so which is covering different topics, medicine, which our institute is active on. Then we have astronomy, geoscience, transportation, and energy. So, and then basically this is combination of different data scientists from the Berlin area coming together, from the Free University, Humboldt University, Technical University, and Charité in Berlin. Yeah, and about myself, I am a junior research group leader at MDC, and I am working, my group is developing computational methods for the analysis of different types of biological data that we know as omics data. This includes genomics, epigenomics, transcriptomics, which is like the activity of genes, and proteomics. So all these words ending with omics things, we are trying to develop new methods for analyzing such types of data.
There we are. Thank you very much, Lale, for this brief introduction into the very, very complex hybrids. I might add to that the hybrids was the very first school that we founded, so you guys are always the prototype that everybody's looking most excitedly about, with 11 partners in all of Berlin, certainly one of our most complex programs, and getting closer to the evaluation. We are very excited to have you with us to introduce the program. So moving to the very north coast, I would love to hear from Anna Biestok about the MAR data.
I'm the speaker of the MAR Data School, and I would like to share the idea behind MAR Data with you. I'm a marine scientist, and exploring the ocean involves a range of disciplines. I'm a physical oceanographer, but there are specialists also from biology, chemistry, geology, all studying the role of the ocean in the global climate system and under climate change. Now, even though these disciplines are quite different, we all share the same challenge, which is a drastic increase in data streams. We have increased data from satellites, deep sea observatories, autonomous robots in the ocean, or high resolution models, just to name a few. And the approach with MAR Data is that we hope to get new insights through data science approaches in our vast range of marine data. So MAR Data, the Helmholtz School for Marine Data Science, is a graduate school within HIDA. And the idea is to aim to define and educate a new field called marine data scientists. And the idea here is that we jointly supervise doctoral candidates, one from the marine side, with all the disciplines I've just mentioned, and then the data science specialist from universities. So GeoMar, the Helmholtz Center for Ocean Research in Kiel, and AVI, the Polar Institute, have teamed up with their university partners in Bremen and Kiel, and jointly educate these doctoral candidates in block courses, summer schools, and colloquia, to get a more systematic treatment of marine data. And we hope to go beyond statistics into really causal relations that we can gather from our marine data.
Thank you, Anna Biestok, for this great introduction. And let me stress the point. If you talk about climate, you have to talk about the ocean. And if you think the ocean is just for sailing and fishing, you're vastly mistaken. And I urge you to check out the MAR Data program. So next in line and concluding the sixth of our 6 great schools, I'd love to hear from Alke Meens what's going on at the DASH in Hamburg.
Good afternoon. My name is Alke Meens, and I'm heading the group FSBMX here at DESY. And this stands for Biomedical Research with X-rays. So we're doing biomedical research using X-rays. And we're having here in Hamburg quite a number of highly brilliant X-rays machines. So this is Petra 3, FLASH, and also the European XRVL. In my group, we are a team of scientists and engineers. We're working closely together, and we're developing new methods for structure determinations from biological samples, making use really of high throughput approaches using robotic arms and machine learning for data evaluation. So, for example, most recently, we have applied this kind of methodology we have developed to a pharmaceutical screening project with the goal to identify drug candidates against SARS-CoV-2, so coronavirus infections. And there we have, for example, screened here at Petra 3, 8,000 individual crystals and took data from them in a big effort. And as you can imagine, if you have 8,000 from 8,000 samples, complete diffraction data sets, this is enormous amounts of data we produce here. In general, in Hamburg at the sources, you can really produce more than 1,000 high resolution images, diffraction images per second. And so this certainly requires efficient automatic data processing. And also later, if you have the research analysis with, for example, machine learning approaches. This is why I'm part or member of DASH, and this is the Data Science in Hamburg School. It's also a Helmholtz Graduate School of Meta. And currently, DASH has 9 member institutions. Among them is DESY, with also strong involvement in particle physics at CERN, the European XRVL, Herion, and Geesthacht University and Hamburg, and other partner institutions. DASH acts as the interface between data science and a certain specific kind of research field. And this ranges from particle physics, accelerator science, over material science to structural biology. And we currently have 24 PhD students, and they are supervised by 70 researchers and also leading researchers, numerous junior scientists from the different fields, and they're also co-supervised. So ideally, there's one expert from data science and one from the specific topic. And at the moment on this booth, you're welcome to visit. We offer 14 PhD positions for research projects.
Amazing. From powerful lasers to single cell and molecular medicines to the vast oceans of the Earth. I mean, I've been working with you guys for 5 years and I'm still blown out of the water by the vastness and the multi-tiered program that's coming together here. I'm truly amazed. Thank you so much for the introduction. Guys, let me remind everyone again of the Q&A on the side, because otherwise it's just me asking stuff and I actually know what they're doing. So. All right, but let's dive a little bit into the programs that you are presenting. Let's zoom in a little bit. In the last session, we got some insight by learning about like a typical PhD project that's been conducted in the school. Do you have something exemplary, like something that you are really excited about, or something that you could explain a little closer to the audience, like a project carried out by a PhD currently? Does anything come to mind? Lali, you want to start out?
Yeah.
Like what kind of project would I be working on if I would apply with you right now?
Yeah, so I introduced these omics data types. So these are the different molecular levels that define the state that each cell is in and is behaving. So one of the very central questions that, for example, in my group or some other computational groups at our institute are trying to address is the relation between these different layers of information, how the information is flowing over these different layers, how they talk and interact with each other, and how all of this in the end governs the cell's behavior, whether it is in development, that we have transitioned from one cell state like the stem cell to more mature cell types in the body, like blood, muscles, skin, whatever, or whether it is the cell state transitions in disease and progression of disease. So we are using all these different information that we can gather from these different types, try to put them together and get a more complete understanding of how cells are behaving.
Very, very, very interesting. Thank you so much. I would love to get into that in more detail. But let's hear from Anna Bierstock. What would be a typical PhD project with the MAR data?
Yeah, I can answer that from my own profession. I'm a physical oceanographer, and I'm running ocean models to explore the role of the ocean currents in climate change. It's fascinating on one side to see how far we came. We can put next to the observations our model results and be astonished how realistic these are. On the other hand, we now have hundreds of terabytes of data. And it's more and more difficult to explore these with traditional methods. So one PhD topic or doctoral topic that I will start in a month from now with a student is to dig into these data and find the climate relevant signal. The idea in the end is to apply that to the sparse observations that we have and to see how far we can identify climate change in the ocean.
There we go. Thank you so much for bringing this to our attention a little more graphically. Same goes out to Hamburg. What would be a typical PhD job to be carried out at DASH?
Protein dynamics, this is what we are currently working on. This is a pretty exciting field. So you probably have all heard about AlphaFold. So based on machine learning approaches of the static structures, which have been kind of determined in the last 2-3 decades, you can now nicely predict all the ground state structures of proteins. So this works pretty well with Google and Rosetta and so on. So this is relatively well understood. What is not understood is then when it comes to more complex interactions. So this means interactions between proteins, for example, and drug candidates on one hand, and the other topic is protein-protein interaction. And to study this, we are now following a massive data approach. So we are really doing this, what I mentioned, this large screening, kind of to create a databases and then apply machine learning to predict this binding behavior. And in order, this should help a lot, for example, for future drug development. So if you kind of really can tailor your drug according to the protein, because now with the static picture, often this predicted binders fail because there's no dynamics involved. And this is one project we are actually currently running together with the bioinformatics group in Hamburg at DASH. And this is pretty interesting. Yeah.
Perfect. But I guess this position has already been filled, yeah? So, but I can apply for something similar. Great. Okay, guys, there's one issue that I would love to discuss with you guys. Last time we talked about the supercomputers, but now let's talk data. I mean, we learned kind of like on the lines during your introduction that your schools not only offer great positions and a great scientific environment, but also very unique data sets. Could you maybe comment a little bit of the quality of the data? Where does it come from? What is it? Its unique structures or sizes? Is there something with regard to the data that you acquire, that the data treasures that at your disposal, or that could be utilized by students joining your program? Okay, Anna, you want to start?
Yeah. The main the unique selling point for marine data is a vast range and a large heterogeneity. Data are quite different. Sometimes video streams, sometimes current data from the deep ocean, sometimes satellites, which are vastly available. Omics were mentioned as well, are an important role in our institute, and model data. Now, these come from different disciplines, and the uniqueness is to bring the data together and make use of it, because it's also clear that we have much more data than manpower to analyze these data. There's lots of data in our archives which cannot be explored because it's so demanding in terms of calibration and in terms of comparison, and make use of it.
Well, I always had the feeling that this is a very, very strong selling point. I mean, all the guys tuned in now from computer science departments who have to work with Monte Carlo data or some sandbox randomized stuff. I mean, hearing about those data treasures is something that I think is particularly exciting. So what about Dash? Do I get some great data access there as well?
A pretty broad range of data. So this ranges from particle physics, where you kind of have continuously stream data from these collision experiments, and you need to filter them and find the signal in the often weak signals in the noise. This ranges to microscopy images, could be, for example, electron microscopy or X-ray microscopy, where you use a coherent X-ray diffraction and collect the data. Then you need to kind of retrieve the phases and so on. So this is talking also about terabytes collected easily in a few minutes or something, and then ends up in my kind of business, which is this crystallography, where you also record diffraction images if you do it time resolved in also, as I said, 1,000 images per second or something. And they need to be also modeled and also kind of first sorted, then modeled. And then you have to kind of in the end retrieve the shape of your sample, or lately identify in some electron density cloud from which you yield as a result from the experiment, identify, for example, drug candidates binding to this. So it's pretty broad, and lots of data. And I think with this, we're aiming now to make this even faster and faster. So to collect for more, to provide better databases also for this machine learning approaches. And with this, we definitely rely on fully automatic data processing. This can't be handled manually at all anymore.
And the machine for the collection are actually on site, right? So at your disposal.
We have a few on site, but we're also kind of internationally. So we're also involved in Stanford at LCLS or Argonne in close to Chicago in the US. So it's quite international, our business, at least the X-ray business. CERN for sure as well.
Fantastic. So, yeah. Lots of data treasures at Dash as well. Lale, what kind of data would I be working with if I would join your group?
Yes, so we are working with this molecular data that I said gathered from single cells. And this is a rather very new technology, not more than 20 years maybe. So before that, people were able only to measure the molecular properties in a bunch of cells, so in a handful of cells. But with this technology, we are able to do these measurements at the single cell level. And that is important because like in some tissues, if you consider brain, many different cell types and neurons are sitting close to each other. So it's not helpful to have an average of a population. So we work with single cell data. And then because the number of molecules that we start with in the single cell are very few, then we have a lot of noise and missing values because we could not capture all the molecules or information that is there because of starting with low molecule numbers. So we have very high dimensional data. So many regions in the genome that we are interested in to look at. For example, if we talk about number of genes, like genes activity. So for human, we have more than 20,000 genes. And then we are working with very sparse and noisy data. And also a lot of this data, we don't know what it is about, right? So the DNA, only 1% of it is coding for genes that we know their function. So there is a big landscape that many regions we don't know actually what their function and role are, and we are interested to learn about it.
Thank you so much. That's one of the amazing parts. I mean, in my scientific career, I spent years to build something, to collect a little bit of data. You guys have treasures of it lying around, or even more, as Anna put it, that can be analyzed at the moment with the Manpower 10. So we need you guys to join the teams and put that data to good use. So I hear that you're really stirring the attention of the crowd. Victoria, we have some questions in the chat.
Yeah, we have a question from Ajay Kumar, and it's asking, as new algorithms and ideas come up every day in data science, I feel combining engineering science with data science also dynamic. How is it possible to keep up with these changes and decide on the best algorithm?
That's a great question. How do you decide on the best methods? Tricky question, I know, but who wants to give it a go? Anne?
I mean, I'm not a data science expert, and so excuse me. So I rely on my colleagues, on my specialists from the universities. My data educates data science, marine data science, but it's important to say the education, the doctoral degree is not in marine sciences, it's in data science. So therefore, if you start and work in MarData, you have to develop new tools and to explore them. You apply them to marine sciences. So that means in the end, you are at the forefront of data science. And of course, I mean, we as marine data scientists, we also learn about data science. MarData also helps us to dig into these topics so that we learn more and more. And our hope is that we have also a nurturing of marine data science into our, let's call it traditional scientists at the institute.
From your answer that it's really important to have a large and vivid ecosystem around yourself where you actually have people toying around with the new techniques so you can decide as a team how great. I think that's a very important aspect of these research schools as well. Are there more questions in the chat?
Yeah, we have a question from Dina Shah. And it said, I got a bachelor in genetics and a master's in bioinformatics. And I noticed recently that a lot of research is going towards single cell omics research. Is this where most omics research is headed?
Lali, I think that's for you.
Yes, so this is an cutting edge technology for doing these molecular measurements. I mean, that's not the whole story, right? So I mean, in the end, cells together, it is the collective behavior of many cells that form the bigger scale things that we see, like for example, in a disease or development. So yes, it's true that single cell technologies are very exciting and bringing a new research area which is developing very fast. But that's not the whole story. So other approaches in biology are still very active and very alive and very useful as well.
Thank you for these insights. I think we have time for one quick question.
Yeah, let me check. There is a very specific question regarding the More Data program.
Sure.
How often regularly do you have calls and application for PhDs? And yeah, is it every 2 years?
I think this can be expanded to everyone here, right? So when is the next call for your school coming up?
So I think ours is currently open. So just visit the booth. It's, I think, 14 open positions. So just go there and have a look whether you talk to the scientists, whether you find something interesting for you.
Fantastic. So we can dive right into the dash right now. How about hybrids and more data? When is your next calls coming up?
Maybe I can continue. Our call has just been closed. We have hired now something like 17 PhD candidates, but the next one is coming up probably early next year, at least at RV.
Early next year, so I have to be patient for a couple of months, okay, but that's not too far away. Lali, what about Berlin? When can I apply?
Actually, I don't know the answer for this question. So we currently have 24 students in hybrid, but I'm sorry, I am not aware of the next recruitment.
Of course, because me tying together this session is not only to thank you, but also referring everyone to the partner button somewhere here. And there's a special booth for all the 3 schools that we learned about now. And you can click on that right now and learn about the written part of the information. Or you can be patient till 5:30 Central European Summer Time. That's not Fiji time, to go into the live chat with the people from the hybrids, the Dash and the Mar Data. So let me thank our fantastic speakers for the introduction of the 3 Data Science School. That was Lali Haverdy from the hybrids, Anna Bierstock from Mar Data, and Alke Meens from the Dash. It was fantastic to have you with us, and everybody who got excited about these fantastic schools, please check out the partner sites and get in touch with the PIs and maybe even apply, as I hear Dash has a call open right this moment. See you guys later, and the next time the latest.